So it's a great pleasure again to welcome the second of our next generation leaders, Lisa Giocomo, who is currently for the past year and a half an assistant professor at Stanford in neurobiology. She started in Mike Hasselmo's lab at BU, where she again had a fabulous thesis looking at rhythms in the hippocampus and what they might mean for coding, and also what cellular mechanisms might be involved in those rhythms. And then she again went to the world-leading lab to study grid cells, the Moser lab, and where she continued to look at a broad range. You know, what really characterizes her work is all the way from channels and rhythms through neural coding and theory of how place cells and grid cells might work. And after her wonderful work, postdoc work, now she has a lab, 5-6 people spanning a broad range, you know, the kind of broad range that we're spanning in the MindScope program, all the way, again, from cellular biophysics through in vivo physiology and behavior and modeling. So I hope we hear about all of that, Lisa.
So thank you for the wonderful introduction, and thank you to the Institute for the invitation to speak today. I had a wonderful set of meetings yesterday. I've enjoyed the 2 talks so far, and I'm really looking forward to the rest of the days today, both and tomorrow. So I'm going to talk today about identifying the ionic algorithms for calculating spatial maps. And if for a moment you think about where you're located in space, you'll recognize that you instantly know your location, both in terms of your global and your local surroundings. And I think we often take for granted the fact that our brain is constantly calculating our location in space. But understanding how and why the brain does this is not a very easy task. So what I want to tell you today is how we're trying to understand the mechanisms that sort of give you this sense of location. So we all know that animals and humans have to be able to navigate accurately in order to solve a wide variety of problems. The real challenge, though, is that to truly solve these problems, what you really need is some sort of internal map of space. And you need this to not only know where you are in space, but you need it to know where you've been and to subsequently plan where you want to go. So my talk is broken into 3 parts today. And in the first part of my talk, what I want to tell you about are some of the cellular or molecular mechanisms that we think might be important for generating this sense of location or a neural representation of space. And I'm going to start by just describing to you how neurons actually encode position. And I'll describe briefly hippocampal place cells, and then I'll spend quite a bit of time talking about entorhinal grid cells. I'll then ask whether or not there might be biophysical properties, so single-cell properties of neurons that could contribute to the encoding of position. And I'll describe to you multiple electrophysiological properties which we've found significantly correlate to very specific features of these grid cells. And finally, I'll ask whether or not we can link these biophysical properties of single cells to the representation of space encoded by cells like grid cells. And in the first part of my talk, I'll discuss some data from knockout mice which sort of indicate how cellular properties might contribute to the neural representation of space. And then in the second part of my talk, I'll discuss how we might be able to use computational models to make these same kinds of links. So the experimental study of spatial memory has a very long history. It dates back to Edward C. Tolman in the 1940s, who introduced this idea that animals and humans are capable of forming some sort of internal neural map of space. And he actually based this work on behavioral studies he performed with rodents. So he would use these complex mazes, and this is a bird's eye view of one of the mazes that he used. And he would have an animal learn the location of a particular reward, in this case up in the northeast corner. He would then introduce the animal to a completely novel apparatus, and the animal would be able to take a novel trajectory, so a novel route, to find the same reward location, even though it had never actually seen this particular maze before. And so the ability of the animal to take a novel route or a novel shortcut indicated to Tolman that they must have formed some sort of spatial map of the local environment, and he called this a cognitive map. Now, the first real breakthrough, though, in our understanding of the neural basis of spatial representations didn't come until the 1970s, when John O'Keefe discovered place cells in the hippocampus. He started recording from cells in region CA1 of the hippocampus as animals explored these open arenas. And what he found was that the majority of cells in this region of the hippocampus would fire only when the animal was in one particular place in the environment. So these are examples of place cells. So these are shown from a bird's eye view, and they're just a 1 by 1 meter open arena, for example. And these black colors indicate the animal's trajectory over the course of, say, a 30 or 40-minute recording session. And a red dot is plotted then every time this place cell fires an action potential. These are just color-coded plots of the same thing I'm showing here. So blue colors in this case indicate locations where the cell did not fire, and red and yellow colors indicate locations where the cell fired at a very high rate. So you can see that this place cell only fires when the animal is in the northwest portion of the environment, whereas this place cell fires only when the animal is in the southern portion of the environment. Now, it remained rather mysterious as to where the spatial information was coming from that allowed these place cells to form such specific representations of the environment. But there was a lot of interest in looking at medial entorhinal cortex. And the rationale behind that was that the superficial layers of medial entorhinal cortex form one of the strongest projections into the hippocampal formation. So starting in 2004 and 2005, Marianne Fien and Torkel Hafting in Edvard and May-Britt Mosier's laboratory began recording from the dorsolateral band of medial entorhinal cortex. And what they found were cells that, like place cells, fired in spatially specific locations, but these locations actually repeated at regular intervals so that over the course of, say, a 30 or 40-minute recording session, this periodic and hexagonal pattern of firing activity actually emerged. And this, because of the periodic nature of this signal, they decided to call these cells grid cells. And I just want to show you a video that was made in the Mosier laboratory of a grid cell. Here you can see this is a rat that's exploring an open environment. And the two little lights are just the tracking LEDs. And every time this grid cell fires an action potential, you'll see a white diamond plotted. And you can see that initially it'll look a little random, but then over the course of the session, you'll see this very nice periodic and hexagonal pattern emerge. And we're just throwing in some chocolate Fruit Loops so that the animal's actually motivated to explore. So now at the end of the session, you can see you have this very nice hexagonal and periodic representation of the environment. And people got very excited about this because it's rather fascinating that a cell is able to actually construct this kind of representation. So grid cells were initially reported in rats. However, they have since been found in mice. They've also been reported just recently in crawling bats. These aren't flying bats. These are actually crawling bats. Just 2 years ago, they were reported in primates, and just last year, they were discovered in epileptic human patients that were navigating through a virtual reality. So this really suggests that grid cells are a fundamental and evolutionarily conserved coding scheme that are utilized by entorhinal circuits. So the very sort of periodic and rigid nature of the grid code has led a lot of researchers to propose that they serve as some sort of universal spatial mapping system for the brain. And if you look at a grid cell, even just by eye, you can see that in some ways they're reminiscent of a longitude and latitude coordinate system. Only instead of a square map covering some sort of two-dimensional environment, the unit of the grid is an equilateral triangle. In order to have this very sort of rigid representation, the grid cell has to be able to continuously correct for changes in the animal's running speed and running direction. And the ability for these cells to do that and have this very nice representation of the environment has led people to propose that they potentially provide the animal with some sort of metric representation of its spatial environment. So there are a couple of features about grid cells that make it an ideal sort of code to cover the spatial environment. First is that as a population, grid cells actually tile the entire environment. So although a single grid cell might have this periodic representation, the next grid cell will actually have a slightly shifted phase so that as a population, you can actually tile the entire environment with your grid cells. And the second is that grid cells actually represent space at multiple different scales. And this representation of space at multiple resolutions or scales is highly topographically organized. So this is a sagittal section of entorhinal cortex. And if you look at grid cells in the dorsal portion of entorhinal cortex, you tend to find grid cells with small scale, so a small distance, say 20 or 30 centimeters, between each grid node. As you move to more ventral portions of medial entorhinal cortex, you'll start to find more and more grid cells that have a much larger scale. So the distance between these nodes will systematically begin to increase. And there's a number of computational models that have utilized the fact that grid cells can represent space at multiple scales to allow downstream structures to actually decode the location of the animal simply by combining or summing different sets of grid cells. Okay, so what biophysical properties or single cell properties could possibly contribute to this very elegant coding scheme that I've just described to you? And why would biophysical properties be important potentially in determining some of the coding features of grid cells? Well, the nature of the grid code suggests that it may arise from processes intrinsic to the entorhinal circuit itself. And that's because these cells actually receive a number of shared sensory inputs, and yet they're able to translate those inputs into the representation of space at multiple different scales. And this suggests that potentially mechanisms for this translation may actually arise from within entorhinal cortex. So as a PhD student in Mike Hasselmo's lab at Boston University, we wanted to ask, well, are there single cell properties or biophysical properties that might change along this dorsal to ventral axis in a manner that's correlative to this change in grid scale? And so starting in 2006 and 2007, I began performing whole cell patch clamp recordings in these layer 2 entorhinal cells. So we ended up finding that there was a number of different biophysical properties that are graded along this dorsal to ventral axis. I'm just going to focus on 2 today. The first are these membrane potential oscillations. So there are a number of oscillatory properties that are graded along the dorsal to ventral axis, and this is one of them. So if you depolarize these cells to near their firing threshold, they'll begin to spontaneously oscillate, and these are intrinsically generated oscillations. They're not synaptically driven oscillations. And if you look at the frequency of those oscillations in dorsal, they tend to be higher than the frequency of the oscillations in ventral. And you can see this very clearly on the scatter plot. Here we have oscillation frequency on the y-axis, and this is distance from dorsal to ventral on the x-axis. And you can see that the oscillation frequency tends to be high in dorsal, where you have these smaller grid cells, and lower in ventral, where you tend to have these larger grid cells. It's not just oscillatory properties that are organized along the axis. There are also temporal and integrative properties that are organized along the same axis. This is an example of how the after hyperpolarization, or the time constant of the after hyperpolarization, is graded along the dorsal to ventral axis. This is an action potential where I've just chopped off the top of it, and the black trace indicates a dorsal cell, with the light gray trace indicating the most ventral cell. And you can see that the recovery after this action potential is much faster in dorsal compared to ventral. And again, if you look at the scatter plot, you can see the time constant for this AHP is much faster in dorsal and much slower in ventral. So we next wanted to identify what the potential mechanism was for these biophysical gradients, and we ended up focusing on a particular current called IH, the hyperpolarization activated cation current. And the reason that we focused on IH was that there was some really nice computational work showing that IH, in conjunction with another current called the persistent sodium current, could generate these membrane potential oscillations. And the general idea is that if you start at some sort of hyperpolarized membrane potential, you'll activate IH, and this will cause a transient depolarization. It's a depolarizing current. This depolarization will activate the NAP, which will actually cause further depolarization. This further depolarization will then inactivate IH, which will cause a transient hyperpolarization. And this transient hyperpolarization will be enough to inactivate the persistent sodium current, causing enough hyperpolarization to reactivate IH. So the push and pull between these two currents can then generate these membrane potential oscillations. In addition, IH is thought to contribute to the AHP. So again, if you have a hyperpolarization after the action potential, you'll activate IH, and it'll help repolarize the membrane more rapidly. So it's perhaps not surprising that we found that there was a gradient both in the kinetics and the density of IH along this dorsal to ventral axis. So the time constant of IH is faster in dorsal, where you see these high frequency oscillations and smaller grid cells, and the time constant of IH is slower in ventral, where you see these lower frequency oscillations and larger grid cells. In addition, there's more IH in dorsal compared to ventral, which is complementary to this time constant change. So what might the mechanism then be for this change in IH and also biophysical properties? Well, IH is conducted by hyperpolarization-activated cyclic nucleotide-gated channels, or HCN channels. And Steve Siegelbaum had done some really elegant work showing that the subunit composition of the HCN channel can determine the kinetics of IH. So if you have a channel that's composed of only these HCN1 subunits, you get very fast time constants of IH. If you have a channel that's composed of only these HCN2 subunits, you get slower time constants of IH. If you have some sort of channel that's in between, then you get kinetics in between these two extremes. So we hypothesized that perhaps a gradient in HCN1 channels might be important for these dorsal ventral gradients in IH and biophysical properties. And to test that idea, we performed whole cell patch clamp recordings in HCN1 knockout mice, as well as their wild type adult litter mates. So here I'm just replicating the oscillatory effect that we saw in rats, which is the data I showed you before. So we found this very nice gradient in membrane potential oscillation frequency in adult mice. And with the loss of HCN1, we saw a profound flattening in this gradient. So either cells didn't really oscillate at all, or they had very low frequency oscillations. On the other hand, if you look at the AHP, you find that there's a very nice gradient, again, from dorsal to ventral in the time constant of the AHP. But when you knock out HCN1, you find that there's an increase in the AHP, but this occurs along the entire dorsal to ventral axis, as there's a number of currents that are actually involved in determining the time constant of the AHP. So you still have this gradient where the AHP is faster in dorsal compared to ventral, but you see this shift along the y-axis. So overall, it's much slower in the knockout mice compared to the wild-type mice. Okay, so before I go on to the in vivo work, I just want to summarize all of the in vitro work that I went through. There are multiple cellular properties that are topographically organized along this dorsal to ventral axis. There are oscillatory properties, and I showed you that the frequency of membrane potential oscillations is higher in dorsal compared to ventral. There's also organization in the temporal spiking properties of these cells, so the AHP is faster in dorsal compared to ventral. And finally, work by other groups has shown that there are integrative properties that are topographically organized. In particular, the time constant of synaptic potentials is much shorter or faster in dorsal compared to ventral. The gradients in all of these properties correlate with this organization in IH. And importantly, the gradients in all of these properties can be either significantly modulated or are lost by the loss of HCN1. So the next question I really wanted to ask was, well, what happens to grid cells if you knock out HCN1? So to answer that question, I moved to the Mosher Laboratory in Norway, and I started performing in vivo single unit tetrode recordings from the medial entorhinal cortex of adult mice as they explored this 1 by 1 meter open arena, which for a mouse takes about 45 minutes. And what we found was that grid cells in these knockout mice were actually significantly larger than grid cells in wild-type mice. So here what I'm showing you are grid cells from different control animals along this column, and these are grid cells in different HCN1 knockout animals along this column. And you can see, if you look at it just by eye, that at each dorsal ventral location, the grid scale is much larger in these HCN1 knockout mice compared to these wild-type mice. You can also see, though, that if you start in dorsal here, you have this very nice gradient where the grid cells tend to be smaller in dorsal and expand to be larger in ventral. And you also see the same thing in these knockout animals. So you see that grid cells tend to be smaller in dorsal and get larger in ventral. And it's very clear if you plot all of the cells together in this scatter plot, you have a very nice gradient in both the HCN1 knockout animals and the control animals, but you have this shift along the y-axis, so grid scale is much larger in the knockout animals compared to the control animals. In addition to these changes in spatial frequencies, there's also changes in some of the temporal frequencies of these cells, and this is just one example of that. In a normal animal, its running speed is actually encoded by the EEG measured theta rhythm. So when the animal runs slow, you'll have some sort of theta rhythm going on. This is the network rhythm going on here. As the animal speeds up, so its speed increases, the frequency of this rhythm will actually increase. And then as the animal slows back down, the frequency of this rhythm will decrease. You can just plot this in normal animals. Here you have running speed from 0 to 50 centimeters per second. And you can see that the frequency of this theta rhythm very nicely increases as the animal's speed increases. When we looked at these HCN1 knockout animals, what we found was that there was sometimes still a little bit of a relationship between running speed and the theta frequency, but in most cases it was profoundly flattened, and this was highly significant. So this really suggested to us that IH may play some sort of role in the gain of the transformation from movement signals to these spatial firing fields. So what cellular properties might actually be generating this effect that I just described to you? Well, I described to you earlier some of these oscillatory properties or resonant properties, and then also these temporal integrative properties, so the spiking properties, and also there's the synaptic properties. And here I've just plotted the oscillatory properties upside down so that you can compare them to the grid spacing. So in the control animals, you had high frequency membrane potential oscillations in dorsal and low frequency membrane potential oscillations in ventral. In these knockout animals, when you knocked out HCN1, you saw a profound flattening in these oscillatory properties along the dorsal to ventral axis. And this really generated the prediction that if oscillatory properties are indeed crucial for setting grid scale, then we would have expected to see some sort of flattening in the gradient in grid scale in these HCN1 knockout animals, and that's actually not what we see at all. On the other hand, if we look at these temporal integrative properties, we have a nice gradient in our wild-type animals, but in our HCN1 knockout animals, we have a significant shift along the y-axis, but a preserved gradient. And in this case, if these temporal integrative properties are really crucial for generating grid scale, then we might have made the prediction that in our HCN1 knockout animals, you would see a significant shift in grid scale along the y-axis, but a preserved gradient. And that's in fact exactly what you see. And so this suggests to us that perhaps these temporal integrative properties are more critical for generating things like grid scale than these oscillatory or resonant properties. So let me just summarize this first part of my talk before I go on to the second part. I showed you that grid cells actually represent space at these multiple different scales or resolutions along the dorsal to ventral axis, and that this correlates with a gradient in biophysical properties along the same axis. I then showed you that HCN1 actually contributes or controls the dorsal ventral gradient in many of these biophysical properties, and that the knockout of HCN1 significantly increases grid scale. So this at least gives us a starting point for trying to understand what the mechanisms are that are generating these cells and their coding properties. So there's a second way to actually approach this, to actually try and link biophysical properties with network coding or with features of the grid code, and that's through computational modeling. And in the grid field, there are two main classes of computational models that have been proposed to explain how grid cells are actually generated. The first are some network level types of models. These models use very specific network architecture in order to generate the grid cell firing pattern. And the second are these oscillatory interference models. And these models use temporal oscillations that are modulated by running speed and running direction in order to generate spatial oscillations. So they basically translate a temporal frequency into a spatial frequency. And I'm going to describe to you a couple examples of these models and how we've tried to integrate single-cell biophysical properties into the models in order to generate predictions and understand how grid cells are actually created. But I just want to emphasize that there are actually many, many iterations of these models, and some of the caveats or challenges that I'm going to describe to you have been addressed by some of the newer models, and some of the newer models bring up new problems and new challenges that I'm not going to discuss. But in the interest of both time and clarity, I'll just describe to you a very small subset of these models to give you an idea of how we can use them to translate from biophysical properties to coding properties. So I want to start by giving you just sort of an overview of probably the most popular attractor model right now. This is a model that was originally proposed by Barak and Fiat in 2009. This is an attractor model. This model uses tonic excitation and recurrent inhibition to actually generate a hexagonal pattern of neural activity. This is an image of the entire grid cell population here. So in this image, each pixel actually represents an individual grid cell, and these cells are recurrently connected. The color code here then has yellow or red indicating high levels of activity, so high levels of neural activity, with black colors indicating low levels of neural activity. And this entire neural sheet of activity will be moved across the population by the animal's movement. So the animal's velocity will actually drive the activity across this neural sheet. And I think it's a little bit easier to understand if I show you this movie. And in this case, we're actually going to be recording from this green cell here. This is one grid cell. And you can see the mouse here is this gray dot. And as it moves around, the activity is going to actually move across the neural sheet. And this results in this individual cell here having a grid cell, having a grid cell firing pattern emerge. And if we were to record from another cell, say over here, we would also see a grid pattern, but it would have a slightly offset phase. Okay, so in this type of model, one of the ways in which you can change grid scale is to simply change how fast this activity gets translated across the neural sheet. So if it gets translated slower, you'll actually get larger grid scale. If it gets translated faster, you'll get much smaller grid scale. And I'll come back to these models in a minute. Okay, well, what about oscillatory interference models? Well, I'll just present sort of a very quick view of the initial oscillatory interference model that I worked with, which was a single cell model. So in this case, you have typically two oscillations that are at slightly different frequencies. That results in the two oscillations moving in and out of phase with each other. And if you generate an interference pattern, so if you just sum these two oscillations to generate this interference pattern, what you'll see is that when the oscillations are in phase with each other, you have something that will cross an arbitrary threshold and generate a spike, whereas when the two oscillations are out of phase with each other, the cell will not cross threshold and will not spike. Now, in this case, you would actually generate some sort of temporal periodicity. So if you just had these two oscillations running at slightly different frequencies, you would generate a cell that, for example, fired every 10 seconds, just based on this interference pattern. But we can actually make it a spatial pattern by simply modulating this frequency by the running speed and running direction of the animal. So if the animal starts to run faster, the frequency, one of these frequencies, in this case the dendritic frequency, starts to speed up. If the animal runs slower, the frequency starts to slow down. And you can also modulate it by running direction. So in this case, the east-west running direction of the animal. So if the animal runs in the east-west direction, the frequency speeds up. If the animal runs in the north-south direction, the frequency slows down. And this allows you to actually generate some sort of spatial pattern. You can get a grid cell by making some very strong and probably incorrect assumptions about the inputs that these cells are actually getting. So if you want to generate a grid cell, you have to assume that you have some sort of cell that has 3 different dendrites, each with a different oscillation, that all summate together with the soma in order to generate the grid pattern, and that each oscillation in each dendrite is modulated by a running direction that's offset by 60 degrees from each other. If you have this sort of setup, then when the animal is in the sort of center of all of these different frequency bands, you have enough oscillations that are in phase with each other that the cell will cross this threshold and fire an action potential. But when the rat or the mouse is outside of those 3 bands, then the cell will not be in phase with the oscillations will not be in phase enough that you'll actually be able to generate an action potential. Now, the reason I present both of these models is that they generate very different predictions about how single-cell properties can actually link to spatial scale. So in the case of oscillatory interference models, they actually use the frequency, or the version that I showed you, uses the frequency of membrane potential oscillations to generate grid scale. And this, in normal animals, actually matches quite nicely with the data. So you can run some simulations of this model and show that you get this nice gradient in grid scale, and the simulated grid scale is actually very similar to what you observe both in vitro in terms of what types of frequency of oscillations you see, and also in vivo in terms of what types of grid scale you actually observe. However, if you look at these HCN1 knockout animals, I showed you that they have this massive decrease in oscillation frequency. And this actually generates the prediction that you should see a subsequently very large or very massive increase in grid scale. And this prediction doesn't hold up at all. So we didn't see anything close to this level of expansion. And it really suggests that this oscillatory idea of using membrane potential oscillations is probably not the most accurate in this particular case. On the other hand, there was a recent attractor model that integrated the time constant of the AHP, and they utilized the time constant of the AHP to determine how fast this sort of neural sheet of activity actually moved across the population of cells. And so in this case, a fast AHP actually results in faster translation of activity, and a slower AHP results in slower translation of activity. And that actually gives a much nicer match to the to both our AHP data and also our in vivo data. So here it's just a simulation that was run by a graduate student in Bruce McNaughton's lab, showing that if the time constant of the AHP changes actually quite a bit, similar to what we actually see in the HCN1 knockout animals, you would predict an expansion in grid scale, but it wouldn't be that large. It would be about 10 to 15 centimeters, which is actually quite similar to what we observed in vivo. And so it's possible that these network models are going to be a better fit or more consistent with the data and a better way to interpret how biophysical properties might be contributing to grid coding features. Okay, so just in the last few minutes of my talk, I want to switch gears a little bit and just talk to you a little bit about how this topographical organization in functionally defined neurons is a more general feature of entorhinal cortex. So not just grid scale is topographically organized. There are more than just grid cells in entorhinal cortex. There are place cells, which are actually in the hippocampus, but in entorhinal cortex, there are also border cells. These are cells that fire only when you're near a boundary in the environment. And there are also head direction cells. So these are cells that act as a sort of neural compass. So they fire only when the animal is facing a particular direction in the environment, regardless of what sort of behavior they might be performing at the time. And these are not absolute compass cells. They don't actually fire when the animal is at true north. It's all relative to some sort of visual cue in the environment. So very recently, we decided to look at whether or not there might be gradients in some of these other signals in the entorhinal spatial circuit. And in particular, we decided to focus on head direction tuning. So head direction cells were first reported in the 1980s, and this is going to be just a little movie that shows you an example of a head direction cell. You'll see a rat sort of running through this environment, and there will be spikes overlaid on its trajectory, and then at the same time, you're going to see this cumulative histogram showing you this is the heading direction of the animal, and you'll see these little red bars appear coding for the direction the animal's facing when the cell actually spikes. You can play the movie. So in the plots I'm going to show you, we're going to basically just take this idea and then we're going to plot it here in polar coordinates. So this is a cell that fires most strongly when the animal's facing the north, slightly west direction. So we first looked at layer 3 of entorhinal cortex. Entorhinal cortex has multiple different layers, and in layer 5 and layer 3 you find the largest number of head direction cells. So we started by looking at layer 3, and what I'm showing you here are different head direction cells from dorsal. Dorsal is actually this first column here, to ventral, which is this last column here. And these are the sharpest head direction cells that we observed. So these are the sort of absolute best head direction cells from dorsal versus ventral. And you can see that in dorsal, you have these nice, relatively sharply tuned cells, but as you move towards ventral, you start to get less and less tuning in your cell population. And you can see this in the scatter plot, so you always have cells that have low tuning, but in dorsal, you have this very highly tuned population, and this population basically falls off as you move to more ventral portions of entorhinal cortex. Again, you can see here, so this is for the most sharply tuned cells. You have very sharply tuned cells in dorsal, and then this becomes less and less tuned as you move towards ventral. And we don't know exactly what the functional implications are for this, but we think that there are probably implications in terms of how attractor models work and how they actually can integrate directional or velocity signals. Now, the interesting thing about this head direction gradient is that it's layer specific. So if you look at the grid cell gradient, it actually occurs across all layers of entorhinal cortex, and it even goes into pre- and parasubiculum. So it goes across into multiple different structures. On the other hand, the head direction cell gradient really only occurs in this sort of wide layer 3 band. If you look at layer 5, you find these extremely sharply tuned head direction cells in dorsal, and even down into 2 millimeters deep, you still find these very, very sharply tuned head direction cells. So this seems to be some sort of layer-specific gradient in directional tuning. Okay, so just to summarize, I showed you that there are these multiple electrophysiological or biophysical properties that correlate to the organization in grid scale. And at least as a starting point, we know that HCN1 contributes significantly to the scale of spatial representation in the brain. And finally, we also know that this topographical organization is not limited to grid cells. There are also these head direction cells which exhibit very nice topographical organization in entorhinal cortex. And we really think that understanding how neural computation occurs in entorhinal cortex, which is located multiple synapses away from primary sensory inputs or primary sensory receptors, could potentially provide insight into coding principles for high-order cortical circuits. So as mentioned, I'm now at Stanford University. Most of the work that I presented today was work that I did with Mike Hasselmo, which was a lot of the modeling and the in vitro work, or Edvard and May-Britt Mosier, which was the in vivo work. But we do now have a lab that we're actively collecting data and pursuing new and exciting projects, and I'd be more than happy to talk to people during the conference about some of the things that we're working on, or we've been working on over the last year or so. And thank you very much for your attention.